Hello, everyone. Welcome to BrainMap, people in the room, and I know there are a lot of people on Zoom. So I'm just going to basically hand it over to our own Dr. Padma Sundaram to introduce our speaker today. But we are very glad that you're here, and just a very small housekeeping issue is that we're not going to have our regular Wednesday meeting. This is instead of that, but I'm happy that you found the room and you're in the right place. So I'll pass it over to you, Padma. Thank you.
So it's our great pleasure to introduce our speaker today, Professor Jeremy Schmahmann. Dr. Schmahmann is the Martha and Robert Fogelman Endowed Chair in Ataxia and Cerebellar Neurology at MGB, where he's the founding director of the Ataxia Center, a center of excellence of the National Ataxia Foundation, and Mission MSA, a founding member of the Division of Behavioral Neurology, director of the Schmahmann Lab for Neuroanatomy and Cerebellar Neurobiology. He's past president of the Boston Society of Neurology and Psychiatry and the American Neuropsychiatric Association, and a professor of neurology at the Harvard Medical School. Dr. Schmahmann completed his medical degree at the University of Cape Town in South Africa, residency in the neurological unit of Boston City Hospital, and a postdoc fellowship with Professor Deepak Pandya in the Department of Anatomy and Neurobiology at BU School of Medicine. His clinical and research interests focus on anatomical substrates of intellect and emotion, white matter pathways of the brain, and the clinical neurology, neuropsychiatry, and systems neuroscience of the cerebellum. He has described the cerebellar cognitive affective syndrome, eponymously named Schmahmann syndrome. He has been recognized with the Norman Geschwind Prize for Behavioral Neurology Research, Lifetime Achievement Awards from the Society of Cognitive Behavioral Neurology and the National Ataxia Foundation, Distinguished Neurology Teacher Award from the American Neurological Association, MGH's Raymond D. Adams Mentor of the Year Award. He has been listed in the Best Doctors in America since 1996. The BrainMap Seminar Series is co-sponsored by the Pre-41 Funded Center for Mesoscale Mapping at the Martinos Center. And that I'd like to welcome.
Thank you, Padma. Thank you, Abbas. Thank you for the invitation, and wonderful to see you all here. Longtime friends and colleagues and some newer faces. And in the time that we have today, I'd like to talk about cerebellum and have an emphasis mostly towards the end of the talk on what imaging has done for us. So in a sense, an homage to brain imaging and the field that you are all pioneering in how it plays such an important role and how we've gotten to where we are now in terms of cerebellum. Just some disclosures, I'll be talking about the atlas, but there's no conflicts. I'm going to start at the end. And basically tell you where we are, and then I'll describe how we got here. So this is our current understanding. There are three major components of cerebellum, the triad of clinical ataxiology: the cerebellum motor syndrome that we've known about for a long time, the cerebellovestibular syndrome, also part of the story for many years, and the cerebellar cognitive affective syndrome. And they have, in broad sense, a localization in cerebellum where the anterior lobe here in the sort of pinky color is predominantly motor, but there may be some cognition in lobule 3. The floccular nodular lobe and part of lobule 7 is the vestibular system, and the rest of the cerebellum is the localization, the representations of those aspects of both anatomical features and function that fall within the domains of cognition and affect. So that's where we are at the moment. Just to help think about how this all could be, the underpinning theory I've postulated as dysmetria of thought. There are many ways to think about this. There are other views about the theory of cerebellum, but the idea is that the cerebellum is an integral node in the distributed neural circuits that subserve everything we do: sensory, motor, vestibular, autonomic, limbic, and autonomic function. And there are two complementary anatomical realities that underpin that understanding. The first is that the architecture of cerebellum is essentially invariant throughout. It's not exactly the same, but it's not like the cerebral cortex, where there is a difference in the cellular architecture in different cortical areas. And the second is the nature of the precision of the connections of cerebellum with extracerebellar structures: spinal cord, brainstem, and cerebral hemispheres. So those are the two architectonic and connectional features that subserve the dysmetria of thought theory. The first is that the architecture being consistent, we think underlies a universal transform. It does something that is constant throughout. The idea is that it makes things better, optimizes performance by modulating behavior around a homeostatic baseline. It does it automatically, implicitly, and according to context. So if that breaks, there should be a universal impairment. It should break the same way. And the idea is that when you damage the motor system, you get the cerebellum motor syndrome that we've known about for 2 centuries. But when you damage the cognitive representation region of cerebellum, you get the cerebellar cognitive affective syndrome. So I'm starting you right at the very end. I used to end the talk with that, but that's where we are. And that's the contemporary understanding, and it's reframed the way we think about not only cerebellum, but the nervous system as a whole. If you go right back to the beginning, the most remarkable fact I discovered only in the last few years is that Vincenzo Malacarne, who described the names of the cerebellum and published his work in 1776, was motivated, as were neuropsychiatrists in those days, by the attempt to understand the relationship of mental function to structure. And his quest was to see whether the number of the laminations in the cerebellar folia correlate with level of intellectual function. So the motivation of Malacarne to help put the cerebellum on the map in the mid-1700s was cognitive. Franz Joseph Gall is a name you know from cerebral cortical connections and phrenology, which took him down the wrong path. But he also made another error, which was helpful in science. He had the sense that the cerebellum was important for sexual function. And he wrote a whole monograph about this, which actually got him banned from Austria. The emperor sent him out because of that. This prompted Florenz, a great name in science in the early 1800s, to do experimental investigations in pigeons where he lesioned the cerebellum and the pigeons couldn't coordinate the flapping of their wings, but there was no loss of motor control. So he thought, and this is the origin, 1824, cerebellum as motor control device is Florenz. He also had the idea that the cerebellum does everything everywhere the same way. In one sense, I think we now think that's actually accurate, but on the other sense, he thought that all parts of the cerebellum did the same functional representation, which would actually be incorrect. Not 20 years later, here at the College of Physicians and Surgeons in New York, Dalton says, I ablated the cerebellum and it lost coordination, but it recovers by 16 days. So the recovery of coordinating power weighs as much against the doctrine of Florenz as the loss of coordinating power immediately after the operation weighs in its favor. Now, no one's ever seen that. It's sort of buried in the literature. But right from the beginning, people are starting to ask the questions about what is going on here. The work of Fulton and Dow and other great names, Berdal, Wallenberg, Chambers and Sprague, all showed us the connections of the spinal cord into the cerebellum. And it broke down into projection to the cerebellum in the anterior lobe in what we call in lobule 8, with the vestibular cerebellum here in the floccular-nodular region, and the rest of it they call the cortico-pontocerebellar system, neocerebellum, with no real sense of what it was doing. And we have this from the work of Snyder, who studies functional topography in the animal model, and shows that there is now, that if you haven't seen this diagram, it's one that you will, should enjoy. The legs are up in the front of the vermis, the arms further back, and the head and neck at the posterior vermis, with the second representation down here in the paramedian lobule 8, but also with projection to the vermis from the visual and the auditory regions. So the functional topography is established, but you'll notice that there's really nothing happening out in the lateral hemispheres. It's like the silent frontal lobe of the old days. All of this, then, the rubber meets the road when you see patients. And so just has one example among many to illustrate that we have to think differently about what cerebellum does, beyond the spinocerebellar ataxias that are very heavily motor related, but more so as well, as I'll come to. So at age 4, this little child had behavioral problems, global delay with speech, language, and learning. And then the brain MRI was performed. They find a pilocytic astrocytoma, which is resected. And then when I meet her 8 years later, here's the old injury to cerebellum. Here's the midline approach to the cerebellum. And this will be getting, as you'll see in the atlas, the inferior region of the vermis and the adjacent deep nuclei. This is a major clinical syndrome in neurology where you damage the midline. At surgery, I'll come to, it's called the posterior fossa syndrome. But she had persistent changes over time with emotional lability, obsessive behaviors, mood swings, behavioral problems, insistence on sameness, all of which led to a variety of psychiatric diagnoses in the old-fashioned model of this is what psychiatry is: OCD, bipolar, ADHD, and autism. But she had very minimal cerebellar motor findings, impairment on some of the cognitive testing. I won't belabor that, but she's having difficulty with her cognitive performance, with her behaviors, and on the scale that's now the neuropsychiatric rating scale, she scores about halfway up on the scale, which is much more than you would do in a healthy population. And so we have then the example of one child who's telling us, I damaged cerebellum and I have all these changes even before surgery of her interaction and social engagement, but also more so afterwards. And so the idea is that the dysmetria of thought damages the cognitive emotional domains in the same way as it does for motor control, as I mentioned at the top. So the cerebellar clinical syndrome of ataxia, the wide-based gait, and coordination of the arms, the speech change, the eye movement change that we were taught by the luminaries of our field is in fact the cerebellar motor syndrome that comes from those areas of the cerebellum and the connection systems that arise from the spinal cord and go back down to spinal cord. That's the anterior lobe and the lobule 8. That is the sensory motor cerebellum, and that's the motor syndrome. So here's the anatomy. I'm going to talk about anatomy, I'll talk about clinical neurology, then get to the neuroimaging. The notion of distributed neural circuits goes back to the 60s and 70s. Dr. Pandya was the pioneer in cortical connections in the contemporary era. Dr. Mechelen put this together beautifully with his colorization of the Brodmann map. The idea is that there are regions of the brain talking with each other, each with their own unique architectural properties that are linked in this precise manner. And then as a consequence of the interactions, the emergent property are the behaviors that we take, whether it's sensory, motor, cognitive, emotional, and so on. The question I came to with Dr. Pandya early in my residency, which is now 40 plus years ago, was could cerebellum be involved in cognition? And we decided to use the anatomical circuitry to question whether the projections to the cerebellum arise from areas in the brain that are not motor related. So if you make injections in the cortex here, you'll actually see the tracks traced through the white matter. This became the subject of the fiber pathways book, which I won't talk about. But you see the projection from cortex through the white matter. Here's the fiber tracks coming down, and they terminate in the pons. Here's the first in the series of papers. You make the injections, and here's the pons represented, and from the ventral view, the different subnuclei, projections from the superior parietal lobule and the inferior parietal lobule, including from areas that are involved with the associative and limbic kind of behaviors, suggesting that these pathways permit the cerebellum access to these behaviors that are important for the modulation and integration of higher order function. When you put a whole series of anatomical studies together over the years, here's your injection and here's your termination in the pons, you see that the connection between the cerebral cortex and the pons, which then goes into cerebellum, arises from all these high order areas: prefrontal, posterior parietal, superior temporal, the paralimbic associative regions, and the parahippocampal regions as well. And they do so with the topographic specificity into the pons. And here, for example, in the prefrontal cortex, each little region has a spray of terminations in the basis pontus nuclei, from where information goes into cerebellum. So that's the one major system. The other major connection is the hypothalamus has all these connections reciprocally with the cerebellum. And the hypothalamus does more than temperature regulation. It's involved in gestation and all kinds of maternally bonding behaviors. It's critical for survival of the species, links into cerebellum in a reciprocal manner. And the feedback from the cerebellum, because I've shown you the feed-forward system, the feedback from the cerebellum, which can be examined not only in single study track tracer evaluations, but with these transsynaptic evaluations that Peter Strick pioneered. We see projections from the dorsal dentate of nucleus and the interpositors to the motor area, but from the ventral dentate to prefrontal cortices. And when you map these together, the motor cortex talks to the anterior lobe and lobule 8, as I've shown you at the beginning, and the prefrontal cortex to the lateral hemisphere crus 2. And in fact, in the dentate itself, there's a topographic arrangement of where the neurons are located that go to motor versus premotor versus the prefrontal cortices. There are a number of other really important circuits that we need to pay attention to that are becoming part of the new technology with optogenetics and ways that we can change behavior in the animal model with the light stimulus. So the ventral tegmental area that I'm sure many of you study projects widely to the limbic cortices, and the VTA has a direct input from the cerebellum. So you can, in fact, change the circuitry and the behavior in the animal model in the limbic domain with social cognition in a mouse by influencing the ventral tegmental area. And the cerebellar granule cells, which constitute pretty much 80% of the brain cells that we have, encode not just movement, but also the expectation of reward. The nuclei are the output system of cerebellum. I'm going to talk about the vestigial because it doesn't get as much attention as the dentate, but it turns out to be really important. And this is beautiful work with the contemporary technology looking at how the neurons are organized at the genetic level. And we see that the different subpopulations of neurons in the vestigial nucleus project to, firstly, get information from different parts of the cortex, the vermis and the paravermal regions and the posterior hemispheres. And they are linked to cerebral cortical areas with a whole range of different functions, not just balance and ocular motor, but arousal, motivation, behavior, and salience. So the vestigial nucleus then, through its different subpopulations, gets information back from the midline, predominant, and vestibular regions of cerebellum to the cerebral hemispheres and to brainstem regions important for behavioral modulation. So that's just a one-liner, a brief view, and there's much more work in the field on the anatomy of the system, but I want to use that as a base to launch into the clinical story. Now, there were reports going back to the 1800s. Combet was the first one I found when I did the archaeological dig in the Cantway Library in those days, when you would actually go to the library and go into the stacks downstairs and get allergy from the dust of all the old articles. But in the 1800s, 1900s to 1940s through the mid-70s, people were describing something going on that's not motor when you have something happening in the cerebellum with degeneration, a focal lesion. But I think it's fair to say that until we actually put this together in a systematic manner, looked at patients who had these cerebellar focal injuries and brought to bear a theory that could at least predict this, all goes to the fact that you need to have a fertile soil to be able to plant an idea and see it flourish. We describe patients who have the lesions and then put this together as a cerebellar cognitive affective syndrome. The proband patient who taught me this was someone I saw in the late 80s. She had a midline tumor that was discovered incidentally when she had a CAT scan because she fell on slipping on the ice. But after surgery, she had a behavioral change. And this is now 25 years old, so I won't belabor it. She perseverates. She has trouble drawing a diagram. She has an agrammatism. She gets her language wrong. And she has a behavioral change that is noted by the nurses in the ward. And if you look at a series of patients, some are doing fine. Here's a Z-score distribution of executive function, spatial cognition, verbal visual memory, and intellectual function. Some people are okay, and some are really not. 3 or more standard deviations below the mean. And the differentiating anatomy was those who were doing okay from the cognitive perspective have lesions that were in the anterior lobe, and those that were not had lesions in the posterior lobe, and those with the most impressive behavioral change had midline lesions. And this is an example that you can see this at the bedside. This is the Ray copy, which is a paradigmatic test of visual spatial organization in a young woman who had cerebellitis. She was treated and got better. And here you see the organization is clean in a figure of similar complexity. And here's a fellow with stroke, and he's asked to copy this diagram. And for those of you who have seen this kind of testing, this is the perseveration that we see in a frontal lobe patient. And similarly, a fellow with a posterior cerebellar stroke has trouble copying a cube, has trouble with the star. He perseverates in this diagram here, and that's his person, and he should have done a better job with that. So just an example of a couple of patients to show what you can see with simple bedside testing, and then explore that with deeper neuropsychology. So the syndrome is a impairment of executive function, spatial cognition, language deficits, and personality changes include blunting of affect, disinhibition, and inappropriate behavior. And this is now the third cornerstone, as I mentioned at the beginning, of the cerebellar clinical phenomenology. You can take this further. So the affective piece is now the neuropsychiatry of the cerebellum, which we think divides into 5 major domains. And you can see why the girl up front was sort of all over the map for the psychiatrist, because she had bits and pieces of each of these. So attentional control, emotional control, autism spectrum, psychosis spectrum, and social skill set comes out when you talk to patients and their families about what is the behavioral dyscontrol that we're seeing in this population. And each of them, like in cerebellum, there's an overshoot and undershoot in the motor control. We hypothesize there's an overshoot and undershoot in behavior in the same way that you have the positive and negative syndrome scale in schizophrenia. There's too much or there's too little. You can also dive further into this, and it turns out that social cognition is an issue, that people have difficulty understanding the intent of others. This gets into the social skill set of the spectrum. And the theory of mind task, which is not easy for anybody, trying to figure out what someone's emotion is by their eyes, you're constrained to 4 choices. As hard as it is, healthy controls get it right. Folks who have autism spectrum do not, and our patients tend to fail that. The same is true in the children who've had tumors taken out. So he has a medulloblastoma, he has the resection site, like our first patient. And this is an interesting hypothesis here, that she copies the ray diagram, immediate recall, and then delayed has this loosening of association, which my student Xavier Guel and I have called phenoconsonance. The same thing in the one system as the other, but tied together by the breakdown in the underlying cognitive paradigm. They have difficulty problem solving, spatial organization, language and memory, and the affective regulation, as I've mentioned above. So that is the essence of the syndrome, which we now have a scale to assess, and so we can do this at the bedside, which is turning out to capture folks who have the syndrome and differentiate them from healthy controls. If you do this in the spinocerebellar ataxias, we find that 40 to 50% of our patients fail 3 out of the 10 domains compared to healthy controls, which we regard as definite cerebellar cognitive affective syndrome. And that's across the board of the spinocerebellar ataxias, type 1, 2, 3, 6, 7, 8, and 10. And they may be early, may be late, but that's the incidence. So this is a real phenomenon in our patients, as reported by them when asked. So the patient-reported outcome measure of ataxia derived from patients, which is now a few years old. There is a mental section, affect and cognition, that makes up about 20% of their burden of disease from the inherited ataxias. And we now have a cerebellar neuropsychiatric rating scale that quantifies and characterizes the deficits I've described in these 5 domains of behavior. So let's jump to the part you're all waiting for, which is the brain imaging. I'm going to do a little sidetrack just for a few slides to make the point that imaging for us in the neuroscience is like the EKG for the cardiologist and the chest X-ray or the chest CT for the pulmonologist. We see them, we study them, and if we don't, we're making a mistake. So the imaging has been enormously valuable across the spectrum, from clinical care to research. And here's an example. This is now 20 years ago, where I explored Dr. Seymour Fisher, our famous stroke neurologist who was here for most of his career, described the lacuna syndromes in the pons. And then I did a very old-fashioned thing with Jason McMore, my lab manager, who we basically made up lesion-based voxel symptom mapping with Photoshop. God love them. You know, each of these was a little pixel with a voxel with pixels in them, and we then compared the location of the stroke with the anatomy, and using the old-fashioned anatomy with diffusion imaging, we're able to come up with the topographic map in the pons, human pons, rostral of the different locations of the facial paresis, the ataxic arm, ataxic leg, the gait impairment, and then speech issues. And it turns out that the rostral lesions were the ones that produced the higher order impairment. So that's one example of the imaging really driving our understanding. Here's a fun one. This is an important sign, being able to identify, looking at the image, that this change in the signal parameter on flare or T2 or dark on T1 is essentially diagnostic of progressive multifocal leukoencephalopathy. There's only one disease that looks like that if it's symmetric, which is an inherited disease called Faxtas. And then 2 more. This one here, looking at this old-fashioned, bringing up, old-fashioned, new-fashioned, bringing up the imaging on EPIC and measuring the pons, we will be able to identify that unlike the inherited ataxias, multiple system atrophy of the cerebellar type atrophies the pons at a rate faster than any other disease. And together with Eva Ratai and Lila Zolay and Andrea Vonikova, in fact, we're scanning people today, looking to see, can we see this change not only over a year, but month over month with the high resolution scanning and using spectroscopy to help us understand the nature of the underlying neurobiology. And then finally, mentioning Eva again, this is an indicator of the role that the Martinos Center and imaging here has played in the national effort to understand the spinocerebellar attachments, both from the perspective of morphology and from spectroscopy. And this paper in last year, but there are many. So this is now already a few years old. This is a beautiful scan from the group here, Bruce, Andre, Otto, Rapino, and John Panameni. And this is now in the new version of Gray's anatomy, looking at the anatomy of the cerebellum, which is so clean on the three-dimensional aspect of the imaging. But with diffusion tractography, pioneered by Van Wedeen, we can see these beautiful images in the rat, monkey, and the human. Here's the crossing of the fiber tracts and the cerebral hemispheres. But in the pons, we can see, here's the, as you saw from the work I highlighted with Emi Takahashi, that here is the pattern of the fibers from the middle cerebellar peduncle into the cerebellum. Rostral pons goes to caudal cerebellum, caudal pons to rostral cerebellum. Work we've known about since Bechterew in the 1880s, but now you see it. That's with you know, old-fashioned myelin staining, and here you see it, and it's really extraordinary high-resolution image of the fibers coming at you, going from pons into the middle cerebellar peduncle, the fibers that are going down. I think it's fair to say that imaging, surprisingly, just opened the gates in terms of the larger science community to what cerebellum is doing. So this is now, you know, this is ancient history, 30 years ago. All right, the early studies of coming out of Mark Rakel at WashU, where they're saying, wow, you can see the brain activate when you do stuff. Here's a name, I'm naming things, I'm coming up with a verb for a noun, and the brain activates like, oh my Lord. This is a cognitive paradigm. This completely threw them. So right from the beginning of the whole of the imaging narrative, the cerebellum was right there front and center. I mean, that's not a subtle activation. And then Julie Fees and Mark Rakel wrote the chapter in our book in 97, showed that the speaking aloud produces activation in the anterior lobe, which is motor, but the verb for non-generation activates a different location. Posterior lobe here, cerebellum right, in lobule crust 1 and crust 2, and lobule 6. Eric Cochin, who was early to the game in autism, talked about attention versus motor system. John Desmond talking about working memory, motoric rehearsal versus finger tapping. I love this one, it's got buried. This is Semir Zeki talking about romantic love, the basis of romantic love. This one disappeared. I'm not sure anybody studied that anymore. But here, you expected anterior cingulate and caudate, and he has a cerebellum. So there was a whole series of studies looking at what the brain is doing. It was like this new tool. Everybody was so excited about imaging, and so they studied everything. And the cerebellum comes up in almost all of them. And the deal with that, the problem that we had at that time was this. Anybody remember this? Do you know what this is? This is the Talairac Atlas. This was the Bible of brain anatomy for imaging through the 90s and early 20s, and late 80s as well, I think. So at the Atlas from 88, but there's nothing, just like terra incognita down here. Grateful to Nikos Makis for showing me that you can do this cross correlation here, but the folks who had already perfected this at that time was in Montreal Neurological Institute under Alan Evans. So I went up to Montreal starting in 1995, and with Julian Doyen, we sat at the console and we identified the fissures to be able to figure out what the lobules were, and as a result of that, produced the MRI atlas of the human cerebellum in proportional space. So you could then use that in imaging criteria. So here you see the fissures, here are the lobules, beautifully seen in three-dimensional space. And then we put this together in a more comprehensive version in the atlas, where we resurrected the nomenclature of Olaf Lassell, who had gotten lost. The names of, you may find names of hemispheres, superior, semilunar, lobule, drove everybody crazy, and they were always butting into each other based on different nomenclatures. Lassell figured this out, and we took that and reintroduced that, and now this is the way we think about cerebellum. The different lobules and how they relate to each other across the hemispheres, with the deep nuclei situated here as well. And then when I was about to finish up, Vern Cavness, God bless him, said, well, where are the nuclei? And I said, oops. So we worked together with Arthur Toga and John Mazziota in UCLA, and we used the early iteration of the cryosection human brain, mapped that into the atlas, and now we have where the nuclei are in the atlas as well. As a consequence, you can then take a study like this that shows pain activation here and the anticipation of pain there, and you can map where it is in cerebellar lobular space. Worked with Nikos and with Vern and Dave Kennedy to look at the cerebellum here and then divide it by these different splines into vermis, intermediate, and lateral hemispheres to find ways to start to unbundle cerebellum in a more comprehensive manner. We could identify, for example, the stroke I showed you earlier, where it's located in the coronal section and on a flat map of the cerebellum. There's many different flat maps, as you'll see as we go through there. Everybody just kind of disagrees with each other. This is the early version, which I published as an abstract, and they refused to take it as a paper, so it lives as an abstract. The sort of slug-like cerebellum looks like that, and end up with a little pump in the middle. But when you mapped at that time already in the 1990s what was available onto the atlas, you see a clear difference between motor function and mental imagery of motor function in yellow. And here's language processing, which is what I showed you in the original work of Lobular 6 cross 1 and cross 2. And then you can see the different tactile maps. For the blue is the foot up the front, hand is the back, and the secondary representation down here in lobule 8, abutting on lobule 9. Based on the atlas, Jorn Diedrichsen developed a probabilistic atlas, which you probably know about, called the SUIT, the Spatially Unbiased Atlas Template of the Human Brain, cerebellum, and it has a deep nuclear component as well through the work subsequently performed. Using the atlas and the development of a new tool called the Activation Likelihood Estimation Software, Catherine Studley, who is now doing beautiful work at American University, performed a meta-analysis of studies that have been done to date. So this is an updated version of our first crude map, mapping motor system in the anterior lobe in lobule 8, language processing, spatial cognition, and working memory bilaterally in different locations. This, in fact, I think was the first study looking at multiple different paradigms in the same person at the same time. So to try and get a sense within individual topography, as seen here in a single case study, that these are located differentially in the cerebellum, depending on your study. And here's a group result showing the same thing in 9 subjects, and here's a nice representation in 3D. Xavier Guell, now at NYU, took the work further that Randy Buckner had shown so beautifully, with taking his 7-state intrinsic connectivity networks that show that the motor system talks to the anterior lobe in lobule 8, which is now old friends to you, whereas the other networks, the default in red, and the frontoparietal in orange, and the dorsal and ventral attention networks all map to the posterior cerebellar hemispheres. And so most of the cerebellum has nothing to do with motor control, even at the level of intrinsic connectivity networks. And then what Xavier did was use the Human Connectome project to look at task activation and resting state to show that you see, in fact, the primary, secondary representation of motor, but also you can find a primary, secondary, and tertiary representation in some of the cognitive paradigms. So that's been looked at again. He also looked at the functional gradients of the cerebellum, which has been done in the cerebral hemispheres, to go from task-specific to task non-specific. So here's the motor regions in the blue, sensory motor regions in blue, and here are the more high-level regions involved in cognitive processing. And he goes from task-focused here in blue through the task multimodal to the task unfocused as in default network, both in the task space and resting state network space, and functional gradients have become popularized in a variety of different areas. And then looking at the nucleus, not to forget about the deep nuclei, this is Strick's diagram, this is work of others. You can see that with DTI, the projections from the cerebellar lobules to the nuclei, the dentate nucleus here, whoops, pick up again. This is the anterior lobe over here in blue. There's hardly any representation, a smidgen in the nucleus, and the rest of it is coming from lobule 6 cross 1 and cross 2 in the dentate. And using resting state connectivity, using the Connectome project, Xavier and the team again showed that there's a topographic arrangement of the functional territories from the default mode here in the red to the visual spatial in the green, and the sensory motor and salience regions in the blue. So within the deep nuclei, and I'm using dentate kind of broadly here, maybe it's dentate. People don't think about the others clearly, fastidial, interpositus, and dentate, but there is a clear differential arrangement of the connectivity within the deep nuclei, mapping onto what we know from the cerebral hemisphere, from the cerebral cortex, connections to cerebellum. So the cerebellar cortex through the nuclei to cerebral hemispheres passes through the deep nuclei, and no surprise, they are functionally organized as well. Now, just going back to the clinical story, and again for a moment to highlight the role of imaging. Here's the anterior lobe stroke with diffusion. Here's a posterior lobe stroke. This is the location of where these are supplied. This is superior cerebellar artery causing that stroke. This is the posterior inferior cerebellar artery causing that stroke. This one produces ataxia. This one doesn't. So what you have then explored more fully in the lesion symptom mapping study now 10 years ago is if you stroke out the anterior lobe, you get the motor syndrome. Through lesion symptom mapping, you have a stroke in the posterior lobe, you have no motor problem, you have the cerebellar cognitive affective syndrome. So we have this convergence of information across the fields from anatomy, imaging, and humans, and of course, there's a whole field of behavioral studies and animal models. While we did a few different studies and a few subjects, this has gone viral in a way. I mean, they're putting people in the scanner 10, 20 times. They're doing a whole range of batteries, and they're coming up with maps. So this is Maeve King, and it's been done by others as well. This is Wayedal. Here's the maps. Here's the motor system in the green. This is all the other representations of cognitive processing. And if you look at the inter-individual variability, it's there. You'd expect it's like fingerprints, but it falls with an overall pattern, which starts to fill in all this white stuff from Fulton and Dow and from Snyder going back a long time ago. And then here, just to pay tribute to Heidi with her work on Alzheimer's, we thought about what's going on in these major neurodegenerative diseases in terms of cerebellum. And it turns out that earlier we had this identification that there's a network selected vulnerability of the cerebellum in different diseases, and depending on where the change takes place in the cerebral hemispheres, so does it have a topography in the cerebellum as well. And so we have the sense that the cerebellum is maybe not just a bystander in Alzheimer's disease, and could it be involved in compensation? Could some of the behaviors that we see in Alzheimer's in fact have a cerebellar signature as well? Not memory. So the CCAS is specifically not an amnestic dementia. Antegrade amnesia doesn't belong in the cerebellar camp. That's not what cerebellum does. But you can use these patterns now to start to intervene. So this is now 2010 we did this, I think. 10 is this is subsequent. I'll come back to the study in 2010. If you stimulate the vermis, you change the default, you change the dorsal attention network. But you stimulate the CRUS1-2, you change default mode. And then this interesting study that Faranak Farzan did was what these colors indicate. This is the gamma oscillation spectrum. And where the vermis versus CRUS1-2, you produce changes in different locations, but what they're doing is you're controlling the temporal dynamics of the network. So you're doing the same thing to different places. That's the theory. It's the same idea in different places. The universal transform topographically organized. And so in 2010, we did the study, which has been replicated by others now, where in schizophrenia, you target the vermis, which is the limbic circuitry, to see can you produce changes in the neuropsychiatry of a schizophrenia patient who's having a lot of trouble. And we found that there were improvements in the negative part of the PANS, which is hard to improve, depression, happiness, sadness, and alertness, and a continuous performance test. And there's now a field of a TMS in neuropsychiatry as studied in the recent Journal of Cerebellum. And then I'm going to finish off the clinical story and the imaging with pediatric neurology. The posterior fossa syndrome is an example. I mentioned this at the beginning with the child with the tumor. It's a dramatic and horrible story. When a child comes in for a tumor resection, they take out the child, the tumor's taken out, they're fine for a day or two. It's really quite remarkable. And then they shut down. They stop talking, they go mute, they're kind of passive, don't respond, they have to be fed. When they get back, they have oral apraxia. They can have a wide-ranging behavior from aggressive and irritable to kind of whiny and passive. And along with that, when that recovers, as the speech always does, they then have the cerebellar cognitive affective syndrome. They can have cerebellar motor syndrome. And because brainstem can be involved, they can have cranial neuropathies and corticospinal findings. And the anatomy, when you look at the series as a group, is localized to the cerebellum midline. It's the vermis, and the efferent tract through the superior cerebellar peduncle, which are heading towards the medial dorsal thalamus and anterior thalamic nuclei, which are all cognitively relevant. Beautiful work from Aaron Bose's group, who was here as a resident some years ago. Now, the cerebellum just puts pedal to the metal when it's developing throughout the trimesters. Here we see cerebellum, like cerebrum, increasing in its size towards the end of the pregnancy, but it continues to grow post-conceptional weeks. Here we are, keeps going, and in fact has more increase relative to brain volume than everything else. This has relevance because when you have a cerebellar hemorrhage in utero, as was shown by the Laparopoulos group in Audiodyplacea some years ago, is that in addition to neurological elementary trouble, they have a variety of language deficits, they have a variety of behavioral deficits, and autism scores in more than 40%. This is real neuropsychiatry of a cerebellar hemorrhage in childhood or cerebellar stroke. And this one here that we're actively working on, this is so prematurity as demonstrated here, 24-week delivery twins. They have had motor, cognitive, and social emotional challenges throughout their lives. They're doing great and they're living their lives, but they are requiring intervention to address each of these. So the one twin, this is age 1 in MRI, and age 14, it's shrunk maybe. Age 4 and then age 14 has shrunk. And the question that's put to us is do these kids, have they inherited a gene that's causing this over time? Or is this in fact the prematurity that's leaving its trace as you scan them over time? This is relevant because when you damage the cerebellum, here this is called a disruption, a cerebellar hemorrhage on the one side that leads to change in connectivity to cerebral hemispheres. A lovely study showing, again, from the group at Washington National, that the motor control changes have sensory motor cortical volume loss, autism has dorsolateral prefrontal, and cognitive and expression has change in premotor and midtemporal cortical volume. So you can tie in where the cerebellar injury is to where the changes are on the cerebral hemisphere and what the behaviors look like because of the circuitry that ties these two together. So if you look at the anatomy that's continuing to evolve, lovely work from Emi Takahashi some years ago, here from 17 weeks and here in the adult, you see a progressive elaboration, as I showed you before, of the middle cerebellar peduncle fibers that lie within it going to where their destinations are, number one, and number two, the radial pathways, which are developmental, disappear and get replaced by the horizontal pathways that are more reflective of the parallel fibers in the cerebellar cortex. And here from 17 weeks to 38 weeks, a progressive elaboration of the middle cerebellar peduncle, which conveys information from the pons, which gets input from the cerebral hemisphere, and also changes in vermis, which you expect over time. So with Lila Zolay and Andrea Gabanikova looking at these folks, actually this is Lila's work, you can manually segment the cerebellum at age 16, but it is tough when they're little. So this is this is a good stab at it, but there's going to be lots of room for error there. And in fact, when you look at twin 1 versus twin 2, twin 1 who's scanned at age 1, it gets bigger when the child is 11. And then maybe a little down, but is that the same or is it different? And then twin 2, more volume at age 4, but less at age 11 and 14, but about the same. So in fact, what looks like a shrinking cerebellum may be a reflection of the fact that the cerebellum continuing to grow doesn't. And you're seeing, in fact, the consequences of what happened before. And that really matters, both for understanding how brain is organized and for talking to families about what's happening on the imaging and what's happening to your child. Now, we're looking at this further. Here's the manual segmentation on different subjects, and then using the Developing Human Connectome project, identifying this is the vestibular cerebellum, this is the anterior lobe, and this is the cerebellar posterior lobe, seeing a progressive change over time from 27 weeks to 45, trying to understand what is the normal development. This is an unsolved question across the age spectrum from birth until later on. And then we're heading to the end. We'll have time for questions. This amazing work with polarization sensitive OCT by Hui Wang and the group here with Lila and Bruce Bishop. This is the polarized sensitive OCT of the block face of the brain. And here you can see the molecular layer, granular layer, white matter, and the composite as identified, and then the three-dimensional representation, the crown and the depths of the cerebellar folium, the surface maps of the layer thickness and the optical properties. And here is a cortical layer thickness sampling in 5 cerebellar cases. So this level of detail, the granularity that we can start to bring to bear to the cerebellar organization really takes us to a new place, which takes us back where we started from. So we had the atlas, which I worked on, 1995, is now 30 years ago, and now with Mrs. Martin Serino's atlas with the extraordinary high-resolution imaging. Here's you can see at the level of the folium, and then what John Samuelson with Bruce Rosen and Marty Ireland have done is develop this digital anatomical atlas at a subfolial resolution where you can start to see little subfolia on the MRI. And I expect that as we get more information about cerebellar functional or organization, its functional representations, its role across the spectrum of neurology, neuropsychiatry, behavioral neurology, these will start to become relevant. It's almost like pruning of dendritic spines. I mean, could it get to that level? You start to look at subfolia in different lobules. Do we start to see some manifestations that are important both for brain organization and for clinical relevance? But we don't know the answer to that yet. But this is the kind of technology that I think helps us get there. And it's a remarkable overlap. I think it's conceptually the development of technology, the microscopes get better. So the development of the technology allows us to do all of this. And we've got Anastasia who's doing her work with this amazing development of technology. That technology allows us to ask questions we couldn't have possibly imagined before. And so it becomes a symbiosis of the technology and the questions. And that, I think, is the essence of what makes it such an exciting place to be. And cerebellum, excuse me for saying it, is probably the hottest, coolest area of neuroscience right now. But I'll end with this concept that the new cerebellar cognitive neuroscience is that it's a clinical triad of the motor syndrome, the vestibular syndrome with autonomic, and the cognitive affective syndrome, which changes the game of how we think about brain organization. The why is the dysmetria of thought theory, and you can think about feed forward and feedback and control systems and all the kind of engineering principles, but there's got to be an overarching view as to how this works. And what I've presented to you is one that I think has withstood the challenges that have been thrown at it so far. How does it do this? The complementary realities of the essentially uniform architecture. There are differences. There's upbound and downbound modules. Those who heard Christopher Zhu talk about this a year or two ago have a sense that there's the Zeeman positive, Zeeman negative bands are relevant, but they all fall within the overarching concept of what the cerebellar cortico-nuclear microcircuitry does. And embedded on that is the topography of the arrangement of connections from the different cerebellar regions to the other parts of the nervous system, all with functional relevance. And this is important to know, both for families in terms of cognition, what's going on with my loved one. It allows us opportunities to intervene and find new ways to treat. And it helps us to think about the cerebellum differently in those vexing neuropsychiatric disorders that have not cleanly fitted the mold so far. So there's a large number of folks through the years who've contributed to the overview that I've presented you, which goes back to the 1980s. Many people here in neuroimaging you'll recognize. I won't read through the names, but between the anatomy and the neurology and the neuroscience, it's been a joy to work with everybody. And I think the TMS is going to be a potential interesting way down the future as well in terms of neuromodulation. I'll end there, but I'll just mention if anybody wants to come to our Texas afternoon conferences, we discuss patients, we talk about the neuroscience of it. You can drop me an email, and we will add you onto the list, and you can join us whenever you can. It's about every other Thursday, and it's patients and neuroscience. So I'll go back to the slide here to keep it up. And thank you so much for your attention. Thanks for the invitation. And I think we have a few minutes for questions.